Thank you. My name is Richard Porter. I'll be speaking on behalf of UniQure today. I'm the Chief Business and Scientific Officer. So, UniQure has a rich history in the AAV gene biology space. We've been working in this space for about 25 years. We had the first approved gene therapy in the world. Some of you may remember Glybera back in 2012. We had the first commercially licensed facility, and we are continuing to develop the first gene silencing approach for Huntington's disease. We also have, with our modified AAV5 platform, a AAV vector that we can dose systemically and not have to worry about neutralizing antibodies, as demonstrated by our Hemgenix, which was a product that was developed in 2017. It was approved by the FDA at the end of 2022 and by the EMA in February 23, with a very broad label with no need for pre-screening for neutralizing antibodies. But that's the past, as was mentioned earlier. In terms of looking forward, we now continue on our site in Amsterdam, which is where the company was founded, and our products continue to use the word AMT for Amsterdam Molecular Therapeutics. The site is the focus of research and development. It's where all the preclinical research occurs. We have a lot of the GNA functions and ADPD and its GMP manufacturing certified. Lexington is the corporate HQ. We do clinical development. It's the location of our commercial manufacturing site. This is where we manufacture the Hemgenix for our commercial partner, CSL, I'll come on to, and the location of most of our quality organization. And more recently, and when I joined, we opened a site in Basel, Switzerland, which is very much focused on the research and development activities. And last year, we hired a new chief medical officer, and this site is growing, particularly on the clinical development side. In terms of our pipeline, the hemophilia B product is approved, as mentioned, and this was commercially licensed to CSL. We continue to do the manufacturing, and CSL is responsible because of their large global and with their franchise presence, they are commercializing the product. We are continuing to take advantage of the AAV5 platform. We have a Fabry product which has a cleared IND and is about to start dosing in patients, as well as other undisclosed projects for the moment continuing in the Amsterdam facility. Our lead clinical program today, which I'll spend some time on talking about, is in Huntington's disease. This is a devastating neurodegenerative disease, and we have an earlier program in the sort of late discovery phase, looking at Alzheimer's disease, focused on ApoE4 homozygote patients, developing a protective form of ApoE. We have two ALS projects. The ALS SOD1 project has an open IND, and that is also looking to initiate patient dosing in the first half of this year. That's targeting the SOD1 gene. I'll talk a little bit about that project. And an earlier project targeting the C9orf72, which, based on some recent clinical data from Biogen, Ionis, and Wave, we repositioned and redesigned our construct to look to address both the sense and the antisense forms of the C9orf gene, given the failures of their studies, and looking to learn very much in the field as to how we design future studies going forward. We also are focused on epilepsy, and our temporal lobe epilepsy project also has an open IND and is about to initiate dosing of patients, as well as expanding in this area, which has many advantages in terms of the clinical development side, which we can talk about. In terms of the platform technologies, we have multiple approaches for the design of the cargo, where we can both express, downregulate, we can do both, we can silence multiple genes and express multiple genes simultaneously, and we are working primarily both on AAV5, AAV9, and modified versions, depending on the tropism that we are desiring in the particular projects. In terms of the lead clinical project now, this is in Huntington's disease. This is a devastating, it's an autosomal dominant inherited disease. It hits patients typically in the 50s, and within 10 to 15 years, there's progressive degeneration in cognition, psychiatric concerns, as well as motor disorders, and after 10 to 15 years, typically death. It's devastating for both the patient and the caregivers to see this, particularly within families given the inheritable trait. Often, the children can become the caregivers of the parents, not knowing if they're carrying the disease. There are approximately 30,000 patients diagnosed with Huntington's disease in the US, and given the nature of when the disease hits, potentially another 200,000. So this is not a ultra orphan, it's a rare disease, but it's hitting a lot of patients. The approach that we are taking in UniQure with AMT-130, this is using a modified microRNA that is designed to knock down the HTT gene. It's been designed to knock down the HTT at both exon 1, and so it will target both an alternative splice version of the gene, which is toxic, as well as the full gene, which also goes on to cause toxicity gain of function. So, and this is delivered in the AAV5 platform. It's intraparenchymally administered. We're basically looking to ensure that the drug gets to the area of need, so we have 2 injections bilaterally into the putamen and 1 into the caudate. So it's quite an extensive surgical procedure driven by MRI-guided delivery with the ClearPoint Neuro device. In terms of the clinical data, we've now we've dosed 29 patients, illustrated on this slide. We have dosed more patients, but they haven't got out to the 12-month time period, so we don't express that data. I'll take a moment to explain this. The top graph shows the clinical scale, the composite United Huntington's Disease Rating Scale. This is a composite score that was developed as a potential regulatory endpoint, taking into account 4 different scales: total functional scale, total motor scale, and 2 cognitive measures. The data is pretty much what you would hope to see in this type of disease, and I'll talk you through it. The circles is a sham-controlled surgery group. This was required by the FDA to allow us to open this study, and these patients were followed for the first 12 months, and after 12 months, if they hadn't degenerated and they met the inclusion criteria, they were offered a crossover into an active arm. That's the circles. The triangles going up is the high dose, and the green line is the low dose. We also have, and this is combined data from the US and a European study. In Europe, we don't have to have a sham-controlled group, so the data is combined. The exciting thing here for us is that we appear to have a dose-dependent effect. As we go out to the longer time points, and this is one of the challenges of neurodegenerative diseases, but one of the advantages of gene therapy is that you do follow your patients for such a long period of time. We can see that the doses appear to separate, albeit with caution, and I actually put on the 2-year, the 24-month time point for the high-dose group. We only have 2 patients that were followed. The next data cut, which is anticipated to be released in June, will be giving us over 10 patients beyond 2 years in both groups, so it'll be much more representative. Both arms track above what the dotted orange line that you can see going down. That is what you would anticipate how the patients would be scored. From there's some very nice natural history data in this disease, and we have done matched propensity matching of patients that would have met the inclusion criteria for the study, and then monitored and seen how they would have compared to our patients. In addition, we have on the bottom panel, the neurofilament light, which is a measure of neuronal integrity. And you can see an initial spike that isn't dose-dependent. That is a consequence of the surgical procedure. And as we get out to the later time points, both arms appear to be tracking below the natural history data for neurofilament light, which again, both scales we consider to be promising and potentially demonstrating an improvement in the clinical as well as biomarker scales. We also have temporal lobe epilepsy. This is AMT-260. We are initiating the first-in-human study imminently. This is designed against a subtype of glutamate receptor. It's, again, a modified microRNA approach delivered in an AAV9 vector. It's an intraparenchymal administration delivered through the occipital lobe into the hippocampus, again using MRI-directed dosing. We have startling preclinical data, which is really encouraging. And one of the challenges we saw using the pilocarpine mice model, which is an extremely aggressive form of bilateral temporal lobe epilepsy, was that the study effectively unblinded itself, and the mice, which were very aggressive, epileptic, they had a lot of aggression, they stopped nesting, they didn't groom. 2 weeks after dosing with AMT-260, they appeared to be normal again. And what we actually found was that these mice had not only a significant reduction in the seizure activity, which you can see in the panel, they also had such a dramatic improvement in their health score, as well as an improvement in induced cognitive decline. We also had the privilege of being able to use resected patient material. Less than 1% to 2% of eligible patients currently go for the standard of care in this disease, which would be resection of the hippocampus, and we can see significant reduction in the epileptic activity of these slices, which are kept in culture for up to 2 years, which retain their hyperexcitability. We have 2 additional studies in Fabry's disease. This is an X-linked genetic disease. There is a Fabrazyme. This project is designed to overcome the PK limitations of enzyme replacement therapy. The patients continue to decline despite the enzyme replacement, largely due to the inability of Fabrazyme to give efficient cross-correction of the disease, particularly in the kidney, the heart, and the brain. And what we've seen with our AAV5 platform, again, is that we get very high and nice and stable, durable expression of the GLA-active enzyme. The ALS SOD1 project is also going into the clinic this year, and that's looking to target a rare genetic form of ALS, building on the success of tofersen, which was recently approved and showing an ASO approach and showing promising data. In terms of our plans for 2024, the Huntington's program will have the next data release, as mentioned, mid-year. We have initiated a 3rd cohort where we will be doing looking at the impact of immunosuppression on the administration, both in efficacy and safety. And we're initiating regulatory activities to understand the potential path to approval. The other programs I've shown you, we are on track to initiate the phase 1/2 studies in the first half of this year, as well as continuing to develop our preclinical pipeline. So in terms of 2024, it's going to be a busy year, particularly in the development area, and we will continue to use our AAV platform to develop the next 4 clinical programs. We're in a strong financial position, fortunately, to take these programs forward into the first half of 2027. And with that, I'll stop, and thank you very much.